So I'm going to be talking about the gram stain. So for the gram stain, we have 4 stains. So CV stands for crystal violet, iodine, which will act as a mordant that will fix the crystal violet, and it produces a complex with the crystal violet, alcohol rinse, and safranin. So we have 2 different types of bacteria, cell walls. We have the gram positive and the gram negative. So when you take a portion of the actual gram, the cell wall of the bacteria, you will have a thick peptidoglycan layer for the gram positive and a thin layer of the peptidoglycan in gram negative. They're both in green. They both have a cell wall. The only difference with gram negative is that they have a lipopolysaccharide layer that gram positive does not. So when you are doing the gram stain, if you were in class, you would have the crystal violet step first. So the crystal violet will actually penetrate the peptidoglycan of the gram positive, and the crystal violet will actually get stuck in the gram negative on the lipopolysaccharide layer. So the next step would be to use the iodine, and this makes the crystal violet iodine complex. Basically what iodine does is when it makes that complex, it's actually called the mordant, which fixes the crystal violet into the cells. So the iodine and the crystal violet will make this complex so that it will not be able to leave the peptidoglycan of gram positive. Since the gram positive, the peptidoglycan of the gram negative is not exposed, it gets stuck in the lipopolysaccharide layer. So when you use the alcohol rinse, alcohol on the gram positive will actually not do anything, and when you have the gram negative, like dissolves like, and when you use the safranin, it will actually stain the peptidoglycan here that is exposed now pink. So gram positive bacteria will be any cocci, so Staphylococcus aureus, Streptococcus aureus, Enterococcus faecalis, so any of those. And also gram positive rods would be any Bacillus species. Whoops. And then Clostridium as well. Gram negative, the famous E. coli, and Proteus vulgaris, and Pseudomonas aeruginosa, which causes burn. Patients that have burns, they are highly susceptible to Pseudomonas aeruginosa. Trying to think. So the gram positive will be purple because crystal violet is a purple color, safranin is a pink color, and the gram negatives will be pink.